Microsporogenesis, that is the development of microspores, pollen, that is the pollen grain. Microsporogenesis is the process in which each microspore mother cell or the pollen mother cells divide meiotically to form four haploid microspores or pollen grains. The microspore mother cells are usually uninucleate. However, occasionally polynucleate condition has been observed in Santalum triticales. Perhaps this condition is due to absence of cytokinesis. The microspore mother cells are interconnected and are also connected with tapetal cells through plasmodesmata. As they enter the phase of meiotic divisions, they lose contact with tapetal cells, become enclosed in thick callose walls, but remain interconnected through bigger channels. Meiosis. The microspore mother cells undergo meiosis, and the meiosis 1 is a heterotypic division and reduction occurs in the number of chromosomes. The meiotic 2 division is homotypic division. Only mitosis occurs. Cytokinesis follows meiotic division of microspore mother cells. The arrangement of pollen grains in a tetrad is affected by cytokinesis during meiosis. And based on cytokinesis, the pollen development can be classified into two types, namely the simultaneous type and second one is successive type. Coming to the first one, that is simultaneous type. In simultaneous type, the cytokinesis occur only at the end of meiosis 2. And this results in production of four cells by cell wall formation between the four nuclei. The resultant tetrads show tetrahedral arrangement and it is common in dicots. Coming to the second type, that is successive type. In this type, cytokinesis occurs twice, once at end of meiosis 1 forming two cells and again at the end of second meiotic division to form four cells. The resultant tetrads show isobilateral arrangement and is found in monocots. Difference between the simultaneous type and successive type. In successive type, each karyokinesis, that is meiotic first and the second division is followed by cytokinesis, that is the wall formation resulting in four microspores arranged in isobilateral manner. Whereas in simultaneous type, karyokinesis, that is the first meiotic division is not followed by cytokinesis. Instead, cytokinesis takes place after second meiotic division resulting in four microspores arranged in a tetrahedral manner. The second difference between the successive and simultaneous type is in successive type, the wall formation is centrifugal, that is from center towards periphery, and this is most common in monocots. Coming to the simultaneous type, here the wall formation is centripetal, that is from periphery towards the center. Arrangement of the spore tetrads. The four pollen grains are united to form a tetrads. Tetrads are unseparated products of meiosis. There are 5 types of tetrads, namely tetrahedral, isobilateral, decussate, T-shaped and linear. And among this, tetrahedral tetrad is the most common. Coming to the first one, that is tetrahedral. The pollen grains are arranged in two different planes. 3 grains are in one plane and one lie centrally over the other 3. In some cases, the pollen grains are released from anther in the tetrahedral condition. These types of tetrads are called obligate or permanent tetrads. present in Drosera rhododendron. This is coming to the second one, that is isobilateral or tetragonal tetrads. Here the 4 pollen grains are in one plane and are equally spaced apart, present in Typha latifolia. Third, decussate. Pairwise of pollen grains are at right angles to one another. Example is Magnolia grandiflora. T-shaped. The first division of pollen mother cell is transverse to form a dyad. The upper or lower cell of the dyad undergoes a vertical or longitudinal division instead of a transverse, yielding either straight or inverted T-shaped configuration. Example, Aristolochia species. Then Polyanthus species. Coming to the fifth one, linear type. The first division of pollen mother cell is transverse and a dyad is formed. Each cell of the dyad again divides transversely to form a linear tetrad. The summary. Microsporogenesis is a process in which the microspore mother cell or the pollen mother cells divide meiotically to form 4 haploid microspores or the pollen grains. Based on cytokinesis, the pollen development can be differentiated into 2 types, namely simultaneous type and successive type. 4 pollen grains are united to form a tetrad. Tetrads are unseparated products of meiosis. There are 5 types of tetrads, namely tetrahedral, isobilateral, decussate, T-shaped and linear. Thank you.